I'm going to be explaining what DNA replication is, what parts go into DNA replication, and why DNA replication is important. DNA replication is the process of replicating DNA. It basically gets one strand of DNA and makes it into two identical strands of DNA used in cell division. There's several vital parts of DNA replication and several enzymes which play an important role into creating two identical fragments of DNA. We first start with the helicase, seen in the diagram as the blue triangle. It basically unzips, like a scissor, the double helix and creates two parts to the DNA strand. The cutting that the helicase does essentially creates some tensions in the original strand of DNA before it's cut. So, to solve this, an enzyme called topoisomerase unwinds the DNA helix and makes supercuts so there's no tensions and no issues before the helicase does its job. Now that we have two strands of DNA, we enter the replication fork, where replication occurs. This is where things become a little different with each strand. This is because of the orientation of each strand. We have one strand that's called the leading strand, which moves from a 3' to the 5' direction, and the other one is the lagging strand, which moves from a 5' to 3' direction. This is because each strand is together in an anti-parallel direction, and essentially this causes a different replication method for each strand. So, now let's move on to what happens in the leading strand. To replicate, we first need a starting point, which in this case is called a primer. A primer is essentially a copy, an RNA segment which binds to the leading strand, which essentially tells the other enzymes where to start replicating the DNA. Now that the primer is set, an enzyme called DNA polymerase knows where the replication method starts. So it just finds the primer and walks down the leading strand, adding complementary nucleotide to the DNA strand, essentially replicating the DNA strand and completing the DNA strand. This is seen in the yellow cube in the diagram, in the leading strand. It essentially walks through the leading strand after the primerase and adds complementary bases to complete the replication process. Now let's move on to the lagging strand. The lagging strand is replicated differently because of a property of DNA polymerase. DNA polymerase can only encode from the 3' direction to the 5' direction. This is the direction of the leading strand. But because the lagging strand is in a direction from a 5' to 3', it must be replicated in a different method. To deal with this, the DNA primase, unlike in the leading strand, places numerous primers throughout the lagging strand, essentially making a primer some empty space, a primer some empty space, and so on. With the help of the primers, the polymerase now understands where to start replicating and when to end replicating, and it now can replicate from the 5' to a 3' direction, opposite to the lagging strand. It adds complementary nucleotides in fragments called Okazari fragments. We can see these on the lagging strand in the diagram. We can see an Okazari fragment in between two primers. This essentially is what I described. The DNA polymerase understands where both primers are, and because the primers are set in different intervals, it knows where to place these Okazari fragments and essentially replicate the DNA. Now that all the bases are matched up and the replication process is sort of done, an enzyme called exonuclease strips away the primers and replaces them with complementary nucleotides, essentially completing the process of replicating the DNA. Now, in the final step, we have DNA ligase, seen in the red cube. What this enzyme does is essentially it joins the two strands together to form the helix. So, in the case of the leading strand and the lagging strand, it joins the old strand with the new strand and basically creates again the hydrogen bond, so that now we have two different double helixes. And the process of DNA replication is done because we start with one DNA strand and now we have two identical DNA strands.